Good afternoon and welcome to this session of Orkney International Science Festival Online. On this closing day, we look at new ideas, radical ideas that challenge established thinking. And the idea of a tree of life is about as established as you can get. The idea that we all diverged from simple beginnings like branches and trees sprouting out of a tree, branches and leaves. And somehow, if we trace it far enough back, the idea is we'll find at the very beginning a single cell that's the common ancestor of us all. But in terms of challenging established ideas in biology, our speaker today has a track record that would be hard to beat. Professor Doolittle has, through his whole career, been prepared to question established ideas and to stand up to the intense debate and criticism that can follow, and eventually to find he's been proved right. His achievements have been recognized by awards such as Canada's highest recognition for scientific research, the Gerhard Herzberg Canada Gold Medal and $1 million prize. He's also recognized as one of the world's leading evolutionary biologists, and he's with us now. Ford, it would be good morning, I guess, in Canadian time. Good morning to you and welcome.
10 o'clock in the morning, so not so bad.
Well, welcome indeed. Can I start by looking back? You've a long connection with Dalhousie University in Halifax, Nova Scotia, which indeed would have begun 50 years ago when you arrived there after completing your PhD at Stanford. I'd like to ask, was your arrival at Dalhousie a time of new beginnings, new directions in your research interests?
Yes, it was, and actually, coincidentally, it was 50 years ago today, exactly today. So that's amazing, actually. Yeah, when I got here, it was a very small place, and I remember when I inquired as to whether or not they'd had experience with postdocs, they said, "Well, we had one, but it didn't work out." and now it's a modern research university, basically. I mean, not my doing, but it really has changed a lot in the 50 years that I've been here. It really was a little college by the sea, which is what it called itself in 1971.
It's wonderful. Exactly 50 years ago. What was the direction that you were going in, the areas of interest before you went there, like in your PhD, and then how did that change?
Yeah, well, I was really supposed to be a biochemist, and I joined the biochemistry department. And I had previously done a postdoc with Norman Pace, who was really important in microbial ecology. And so I was supposed to be doing biochemistry of RNA processing, and I really wasn't a biochemist at heart. I was a molecular biologist and I was an evolutionary biologist, so I quickly diverted from that. And
this was, I guess, this was the time when great developments in DNA were happening.
Yes. I mean, we already knew that DNA was a genetic material. We'd known that for almost 20 years. And we had no idea that you could ever sequence DNA. That was ridiculous. But we had molecular biology, and we also had something called the endosymbiont hypothesis, which Len Margulis was big in promoting, and so that, and her book came out in 1970, which is about the year I came here, and so that converted me to, with some technology that was borrowed from Carl Woese, to try to test the endosymbiont hypothesis, which is what got me into evolution. I wasn't previously formally trained. Well, I've never been formally trained in that field.
It would be interesting to hear more about that hypothesis, because I understand that that's highly germane to the topic of the tree of life.
Well, so Lynn revived ideas that were almost 100 years old, I think. And she published a book which was very influential for some of us in 1970, called The Origin of Eukaryotic Cell, I think it was called, in which she proposed that the chloroplasts were degenerate cyanobacteria that had become endosymbionts within the nuclear component of the eukaryotic cell, and that the mitochondria were degenerate turns out to be alpha proteobacteria now, that were respiring bacteria that had become trapped as endosymbionts within the eukaryotic cell. And this was a fairly radical notion at the time. And Carl Woese had developed RNA oligonucleotide cataloging technology, which was a very tedious compared to sequencing, which now, of course, we do at the drop of a hat. But nevertheless, a feasible technology for testing this hypothesis, which is what I and a colleague named Mike Gray at Dalhousie did. And I think we probably proved the endosymbiont hypothesis.
And just to go into the background, into some of the basics now, the chloroplasts are in plant cells, containing chlorophyll and helping them convert carbon dioxide into oxygen. In fact, I'll just check that with you first.
That's correct, yes.
And the second one then is the mitochondria in animal cells and more like power packs.
Yeah, right. Well, they're also in plant cells, but yeah, but okay. Plants have the chloroplasts as well, right.
So people were looking at those, and initially the idea would have been those are just components of the cells that have developed. But sure. The new hypothesis at this time was these originated outside the cell as bacteria coming up to take residence, a kind of friendly, beneficial, mutually beneficial residence.
Yeah, it's not clear that the benefit was mutual.
I see.
John Maynard Smith, who's one of your more famous evolutionary biologists in the UK, said that early eukaryotes kept mitochondria like humans keep pigs. So, it's not clear that the mitochondria weren't kept rather than benefiting.
In each case, the chloroplasts and the mitochondria, they are doing an absolutely key job that benefits the cell in which they're resident.
Oh, absolutely. Yep. I mean, you wouldn't be here talking to me if it weren't for that. Hold on just a minute here. Yeah, okay, sorry.
And now I know from the DNA studies that are done that there's mitochondrial DNA as well as the DNA in the nucleus of the cell.
Sure.
Was that taken, the fact that there's separate stock of DNA in the mitochondria, was that taken as a hint that indeed they came from outside?
Oh yeah, for sure, yes. Yeah, Lynn Margulis was using that datum as part of the argument.
So that was really the point at which something moving horizontally between cells started to become possible, as against the sheer verticality of the idea of the tree of life with the common ancestor.
Sure, no, and that's true. I mean, one of the largest violations in the tree likeness of the tree of life would be the lateral influence of the mitochondria. I mean, that is a reticulation in the tree, and I think that we dismissed it in the 1970s because we thought, well, there weren't very many genes that came in with the mitochondria, and it wasn't very big compared to the rest of the cell, and so it was a kind of a tiny addition, and that we could track the history of the eukaryotic cell from the nuclear components, basically, and that the mitochondrion was sort of like an add-on. But actually now I think that probably more eukaryotic genes in the nucleus are actually derivative from the mitochondria than were present in the beginning. So it's probably not true.
And just to check again, there's these two types of organisms, prokaryotic and eukaryotic. The eukaryotic are like all of us, a mass of cells with a nucleus and chromosomes and DNA inside, but the prokaryotic ones are really very simple.
Yeah, well, they're not very simple, but yeah, but they are different. They're different in that respect. Yes, they don't have nuclei.
Originally, how did the tree of life look? Because you had these very different kind of organisms. If you could just outline what the conventional picture was when you started looking at it.
Well, I think there wasn't the conventional picture at the time. I mean, I think everybody thought that eukaryotes arose from within the prokaryotes, and that's what Lynn Margulis thought, but by endosymbiosis rather than just straight tree-like branching. But I don't think there was no legitimate way to connect the prokaryotes in the eukaryotic world, which is one of the things that Carl Woese set out to do, was to try to connect the prokaryotes and eukaryotes, because clearly they were connected. There's a unity of biochemistry involved there, but nobody knew how they were connected. And when I was a kid, I think we believed that bacteria, which was the only prokaryotes that we knew at that time, were fungi or something like that. And I think we thought they were fungi. Or plant-like anyway, maybe.
Was the idea of the tree of life almost something that just people absorbed as a kind of hazy general principle, rather than actually looking at detail and what it might mean in practice?
Yes, but I mean, so I think the history that, well, I mean, who knows what the real history is, but the potted history would be that we had classification, and classifications are intrinsically tree-like. I mean, it's really hard to get your mind around a classification that isn't, groups subordinate to groups, right? So that's what Linnaeus's classification was, and one of the things that one of Darwin's accomplishments was to recognize that there was a genealogical basis for that there was a branching process, a tree-like structure that underwrote the classification. And I mean, a tree is a hierarchy, right? It's branches subordinate to branches. So they two go together very nicely. And what people like Haeckel and people in the 19th century and the early part of the 20th century did was basically just take the classifications and assume that they were phylogenies. I mean, so that, a classification was based on non-molecular traits, but similarities and differences. And people assumed, that mostly correctly, that there was a phylogenetic reason for that.
So the concept of a tree led to this idea of a single cell that was the common ancestor of us all.
Yes, I think it did that. I mean, Darwin talked about life being first breathed into a single form or a few, he implied, which I think we could argue was actually a single form, because there's so much similarity between all living things now that it's hard to imagine that they didn't come from some common ancestor that was already highly evolved. So that's what people call LUCA, the last universal common ancestor. Some people debate about whether or not LUCA was a population, but that doesn't make any sense to me, because I can't imagine, because the population would be distributed over time and space, and I can't really understand how that could be an ancestor. It could be an ancestral population, but not an ancestor. And personally, I believe that the last common ancestor that LUCA was a cell or possibly a species if it was sexual.
The classification was originally in these two categories. It was the bacteria on the one side of the divide, and the eukaryotes with the DNA with the nucleus on the other one. But sometime, I think, was it about the 70s or 80s, there became and a third category was brought in.
Yeah, well, there is some history there, and the notion that the world is divided into prokaryotes and eukaryotes is due to Roger Stanier and Cornelius van Niel, who was his boss, and I think that was a structural distinction. I mean, eukaryotes have nuclei and prokaryotes don't, but I think that most people actually believe that eukaryotes evolved from some particular branch of the prokaryotes. So it wasn't actually a phylogenetic distinction. I think that Carl Woese has made it into a phylogenetic distinction because it serves his interests, because he did discover the archaea, and that clearly divides the prokaryotes into two large groups, what he called the mains, the bacteria on the one hand and the archaea on the other hand. Archaea we already knew about beforehand. We just thought they were bacteria, just other bacteria, somewhat weird, and their weirdness is more. They're even weirder now, but still they are prokaryotes. Yeah.
And so there was a division. Was it advances in molecular biology that made it possible to differentiate these two different categories?
Yes. I mean, so Woese set out in the late 60s, I think, to use ribosomal RNA as a universal molecular chronometer, as it were, to unite the prokaryotes and the eukaryotes, but particularly to come up with some kind of phylogeny for the prokaryotes, because nobody really, people had given up on that. Stanier had given up on that because it was impossible, or it seemed, so Woese chose ribosomal RNA, which at that time couldn't be nothing could be sequenced at that time, but ribosomal RNA could be characterized in terms of its characterization depended upon its sequence in such a way that you could actually compare organisms on the basis of that. And all organisms have ribosomal RNA, and furthermore, mitochondria and plastids, chloroplasts have their own ribosomal RNAs too. So that made it possible to test the endosymbiont hypothesis, which is what we did. So yes, Carl and George Fox discovered, his postdoc discovered that archaea and bacteria were two very different divisions within the prokaryotes.
How strange, because outwardly, a lot of them would look fairly, it's not similar, but at least related.
Oh yeah, no, I don't think you could tell the archaea from the bacteria. Well, people didn't before that. They just thought they were weird bacteria, different kinds of weird. Some of them were thermophiles, some of them were halophiles.
When that discovery was made, then that this group had divided into two, how did that affect the way that your own ideas were developing?
Well, I bought into that pretty early because I was a friend of Carl Woese's, for one thing, so I had privileged access to that data. So Woese and would like to have said, he's dead now, that his three-domain vision of things, which was bacteria, archaea, and eukaryotes, the three equally distinct domains, replaced the two-domain vision of Roger Stanier, which was prokaryotes and eukaryotes. But I actually don't think that's the case, because I think we already understood that relationship. But that's a historical issue. But yes, it was quite a radical thing to recognize that bacteria, that what we previously called bacteria, were actually two kinds of things, bacteria and archaea, and there are substantial differences between the molecular biologies of those two different groups, although there's an awful lot of a gene exchange between bacteria and archaea because of lateral gene transfer. So, your average archaeon might not be so different from your average bacterium in terms of about half of its genes.
Thinking now about the concept of the tree of life, I just, when I was reflecting on it, I thought, this is something rather Victorian about it. It's like the family tree, the head of the household, and property going down to the next generation. And I just wondered if there was something rather old-fashioned about it.
Yeah, I suppose there is. I mean, again, as I said earlier, I think that insofar as it underwrote classification, and to my mind, any useful classification has to be hierarchical and tree-like. Yes, probably it also derives from genealogical trees and from patriarchy and all those things, but it also is the easiest way to classify something. So yeah. But that's a historical question. I don't really know the answer to that.
What would you say were the main arguments for challenging the existing theory and changing to a new picture?
Well, I think lateral gene transfer, that Scientific American article you read, which was based on an article in Science that I wrote, but many people were beginning to question the validity of the tree of life based on gene sequences, because I talked about this before, but when molecular techniques became common to do phylogeny with, and when comparison of molecular sequences became the way that you did phylogeny, then in the mid 1960s, Emil Zuckerkandl and Linus Pauling wrote a paper which I thought was very influential, where they said that insofar as the tree based on molecular sequences is the same topology as the tree based on morphological resemblances between organisms, then Darwin would be confirmed. And I think to some extent that's true. And so part of the impetus behind molecular phylogeny was the confirmation of Darwin's claim that there was a tree of life. But part of it was also simply reconstructing that tree of life, because it became by far the easiest and the least arguable way of reconstructing phylogenetic relationships. So I think then people started to assume that, well, it didn't really matter which gene you picked. You could pick any other gene, any gene, basically, because they should all have the same tree, because basically species give rise to species give rise to species, and it's a tree-like pattern that you're trying to recreate. And then so any gene in principle should be able to do that. And then Carl Woese picked ribosomal RNA because that was the most broadly distributed gene, and for many other reasons it was a good choice. But then people started to realize, well, some protein coding genes don't actually show that same topology. And we did some of that too. A guy named Jim Brown, who was a postdoc of mine, published what I think was an important paper showing that of the available data, which were not very many at that time, about a third of them showed the standard tree of life, but a third of them didn't, and a third of them, so the three possible trees were represented almost equally. And so I think people started to realize that, yes, the different genes will give different universal trees, and then when complete genome sequencing became available, and the one that impressed me most was Thermatoga maritima, which is a thermophilic bacterium, and it turns out that a quarter of its genes were otherwise known only in archaea. So lots of lateral transfer, and then more and more data started to accumulate showing a lot of lateral transfer from bacteria to archaea, and from archaea to bacteria, and within the bacteria, and within the archaea, and some into the eukaryotes as well, so that lateral gene transfer became a challenge for reconstructing the tree based on gene sequences. And it doesn't actually mean there isn't a tree. It just means that we can't reliably reconstruct the tree from gene sequences unless we pick a gene and we say that's the tree.
This process of lateral gene transfer, how exactly does it work?
Well, there's a lot of ways. It's a phenomenon rather than a mechanism. Mm-hmm. And so, it's recognized by a particular gene in a genome not showing the same evolutionary relationship that the other genes do, but it can happen for bacteria by the three principal routes: transformation, which is just the uptake of naked DNA, and then there's conjugation, which is a deliberate sexual-like process that bacteria engage in, And transduction, which is a kind of an accidental thing that happens to bacteriophages, viruses that infect bacteria. Those are the three principal mechanisms, but there are other mechanisms that are more recently discovered that I don't know very much about, all of which will have the ability to take a gene into a foreign recipient. And there are many plasmids which deliberately do this. I mean, they take antibiotic resistance genes, for instance, from one species to another species, making it antibiotic resistant, which is one of the problems that we have with superbugs. So lateral gene transfer, and that with lateral gene transfer was actually first discovered as an important biomedical process through the spread of antibiotic resistance in the 60s.
I was just reflecting that the bacteria, we look at them as very primitive, and yet they've got the ability to surprise us in all kinds of ways.
Oh, they're not primitive at all. They're very highly evolved, much more efficient than you or me.
Which I guess is the reason that they've survived such an incredibly long amount of time.
Yes, well, this is largely a bacterial world, after all.
In which we might turn out in the long run to be transient, and the bacteria continue.
We might well be, yes. Yeah.
Just to continue on the development then, with this phenomenon of lateral gene transfer, that then led to you basically rewriting the concept of the tree of life and picturing it instead as a web.
Well, yes. I mean, again, I'm not sure how you turn a web into a useful classification. So I'm not a disbeliever in trees. It's just that I'm a disbeliever in being able to reconstruct the tree from any particular gene. So, I'm sorry, what was the question?
It was just this idea that the model originally was that the tree, and right, it was a very rigid sure, flow, and then that it was more like a web where things were moving around much more fluidly.
It is much more like a web, and there is a much more complicated and nuanced process, which is the evolutionary process, than we had imagined that there would be in Darwin's day, for instance, when we thought about the tree of life as a a monolithic improvable entity. So yeah, I mean I don't really think that there is a tree of life, but so Eric Baptiste and I published a paper in 2007, which I think was a good paper. And Eric is very much of a network kind of guy, and so he really thinks networks are the answer. But I still don't understand how you make a good classification of a network, and how you make a stable classification out of a network. So, but I think that the true picture is a combination of tree likeness and network likeness, and that's what the evolutionary process is about. And for some branches of the tree of life, for instance, the mammals or something like that, it probably is very tree-like, hardly net-like at all. And for bacteria and archaea, for some groups within them, it's probably closer to a web than it is to a tree in terms of the phylogenetic relationships that you might try to draw from them. So I prefer to think of it as a tree with many reticulations rather than as a network.
When you developed these ideas, when you published them, what kind of reaction did you get?
Well, so it's interesting. I mean, I give you a little history that the paper that I published in 1999 in Science was a speculative paper in some ways, because I wasn't sure how serious lateral gene transfer, how prevalent lateral gene transfer was going to turn out to be, and so when I reread it now, I can see how careful I was in wording it. I sort of said, if it turns out to be as serious as some people fear, then it really threatens the concept of a tree of life. But I didn't actually say that it did at that point. And so, but that's an interesting paper, because science asked me as an evolutionary biologist to suggest some people to write articles for them, and I said, why not me? And I guess I think they were a bit embarrassed by this, because they couldn't really ask somebody for their advice and yet refused to accept them as an author. So that's how that paper actually arose, and I got a lot of advice on the writing of that paper. And I'm proud of it now, because I think it really was prescient in some ways, but there were 4 of us who were sort of pushing that same agenda at that time, was often referred to as the 4 horsemen of the death of trees. And then Bill Martin in Germany, and Jeffrey Lawrence in Pittsburgh, and Peter Gogarten in Connecticut, and me. So I think there were 4 of us that were pushing that agenda.
Did you find that having other people that you could bounce ideas from, share information, did you find that helped?
Oh, absolutely, yeah. And one of the papers that's reasonably highly cited, I actually invited. I had some extra money, so I invited Jeffrey Lawrence and Peter Gogarten up to Halifax, and we spent the weekend writing a paper. We just sat in my office and forced ourselves to write this paper in 2 days, and it was actually a pretty good paper. So, yeah, no, I think it was a big help to me having some people who believed the way that I did.
Did you find that, on the other hand, there were other people who disagreed or felt you were even wrong to challenge something as well established?
Yeah, true, and in particular, I think that was probably marked the beginning of my falling out with Carl Woese in some ways. So I don't. He used to send me papers to review, but back when we were friends, and I used to refer to myself as secondhand Woese, but he stopped doing that when I started challenging the tree with lateral gene transfer, because even though he had emphasized the importance of lateral gene transfer early on, but he thought that it had stopped being important evolutionary process prior to the formation of what he called the progenome. Yeah, so there was opposition, and there still is opposition. The view that I currently favor is one that's been being pushed by Eugene Koonin at NIH, which is called the statistical tree of life, which sort of implies that if you pick a bunch of genes, a relatively small bunch of genes that are present in almost all organisms, and you construct a tree yet the trees will be different from each other, but there will be a common thread, a common general consensus view, and that's the statistical tree of life. And I would argue that that's probably the best you're going to get in terms of pattern of the speciation events that are involved. And so you could argue that there's a tree of species, even though there's not a tree of genomes. And I would argue that cellular fusion, or the introgression of radically different species, not recently diverged species, is a rare event, relatively speaking, compared to lateral gene transfer, so that there might be a tree of species even though there's not a tree of genomes because of lateral gene transfer.
Now, a general question. Is it sometimes the case that anything that Darwin said, there are some people who just wouldn't want it questioned, or is there more flexibility?
Yeah, well, I think when you scratch biologists, you'll find that there are 2 types: those who want to prove that Darwin was wrong, God damn it, and I'm right, and those who like me would say, Well, neo-Darwinists are wrong. Everybody since Darwin has been wrong, but I'm right. And Darwin, if he were alive today, would pat me on the back and say, "Yeah, you're right, Ford." And so I think those who seek Darwin's approval, retrospectively, and those who seek to overthrow him are two classes of biologists. And so I'm of the former class. I'm hoping that Darwin would approve of what I'm saying if he were alive today. But
I had a
it's hard to get over Darwin, right?
I had a question to ask about the future, but an interesting one has come in from Bill Graham in Forres, who was interested to hear what you were saying about chloroplasts and the mitochondria. So Bill wants you to look at the future and say, and would it be possible to engineer chloroplasts, which are benefiting the plants from sunlight, and insert them into animals or even human skin cells to add solar energy to the energy provided by the mitochondria?
Well, I suppose so. I mean, you'd have to do an awful lot of engineering, and you'd have development process. I mean, maybe if you don't want this to be heritable, so that it's just a somatic change that you affect on the skins of people, you could probably do that. I mean, it would be a hell of a task to make sure that the chloroplasts actually survived inside the skin cells, which are not designed to do that. But I suppose you could do it, maybe. Yeah.
I should say that Bill, who put the question in, is an engineer and an inventor with several patents. So you maybe have sparked off an idea there.
Well, maybe, yeah. Good luck.
Now, moving to the present time, Ford, an amazing moment again to say, of course, 50 years exactly from starting at Dalhousie. What are you doing now, and are there areas that you're looking to investigate?
Yes, well, I am doing something now. I mean, I've been formally retired for 15 years now, I guess, but I haven't stopped working. And I'm still getting paid a little bit, so that's nice. But I'm trying to. It's the Gaia project. I'm trying to. I was one of the earliest neo-Darwinian opponents of the Gaia hypothesis, because the Gaia hypothesis postulates that the whole biosphere is an evolutionary entity, an organism-like entity, which is evolved by natural selection. And Darwinians, including the ones that I have faith in, would argue that that's impossible. In theory, it's impossible in theory because in order to have evolution by natural selection, you have to have differential reproduction. Basically, the current theories of evolution by natural selection, the ones that I would favor, entail differential reproduction. The thing that reproduces more, the trait which allows it to reproduce more, becomes more frequent in that population. That's the way it works. And the biosphere as a whole doesn't reproduce as a whole. Obviously, it's made up of many species, but they're all essentially selfish in the sense that they are out for their own perpetuation. They're not out for the perpetuation of the biosphere as a whole. So is there any entity that is the biosphere as a whole that is out for its own perpetuation? And that's what the Gaia hypothesis says. And it's what many Darwinians, including me, objected to when James Lovelock published that idea in 1979 for the public. And I've been trying to rethink that, coming up with some mechanism by which the biosphere as a whole could be a unit of selection. I'm not sure I'm going to succeed in that or not, but that's my goal. So we have a bunch of modelers and so I've been trying to do philosophy for the last 10 years now, I guess. Not totally successfully, because I don't I'm not such a slow reader, but anyway, that's what we're trying to do, is to expand the Darwinian worldview so that it includes not only differential reproduction, but differential persistence, because most of the entities that are long living are persisters rather than reproducers. And so it's a matter of coming up with some formulation for that. So that's the goal.
Would you say that the concept, the Gaia concept that James Lovelock developed, would you say that it is gradually starting to be more widely accepted?
Well, okay, it's always been widely accepted by, well, I remember that, so we had Tim Linton, who was a major biogeochemical modeler and a perfectly legitimate scientist who's also been a Lovelock supporter, over to Dalhousie to give a talk a couple of years ago, and one of the audience said, "I thought Gaia was just a hippy dippy idea," and so there is the hippy dippy component of it, the earth goddess type of, which is not what I'm trying to support here. And then the Gaia hypothesis, Lovelock's original work was, to help NASA, or not his original work, but the reason he came to my attention was because he was trying to help NASA decide how we would decide whether there was life on Mars or any other planet. And Lovelock pointed out that the atmosphere would be in wild disequilibrium if life had an input on the atmosphere. And that's true. And so it's been very influential there. And I think the whole system, the whole field called Earth System Science, which is based on Lovelockian principles or Lovelockian perceptions, is based on that on the Gaia hypothesis, sort of, and many people who do biochemical geological processes and who look at biogeochemical cycles sort of think, well, if it weren't that way, it wouldn't work. And so what we're trying to answer is, well, what's the it that wouldn't work? What does work even mean in that larger context? And I think that many people have proceeded to work as if there were an it that worked, and yet not having a sound theoretical justification for believing that there is an it that works. So that's what my goal is.
And just one more question to round off. Again, a more general one, and I'm thinking now of young biologists whose careers are now starting. What advice would you give to a young biologist starting today, whether general advice about practices or about particular areas of biology that you feel that are worth closer attention?
Well, I don't know that, and I would, if I were, I mean, I'm glad that I'm as old as I am, because I wouldn't want to be starting out today because the job market's so terrible, and we've also produced way too many biologists already. I mean, because the field has sort of exponentially grown, and so there's too many of us now, but. I can't but believe that ecology is where it's at. I mean, that understanding the way the world works, if there is an it there, is obviously very important right now, and something that we don't really understand yet very well. So that's what I would recommend, is to combine evolutionary biology and ecological perspectives in some kind of a whole that I'm not yet quite capable of articulating myself. I mean, that's to me what is exciting out there right now. Whether or not there's a career in it, I don't know.
Ford, it's been fascinating and a privilege for us to join you on this special anniversary day for you at Dalhousie. Thank you very much indeed. And you've visited us before in Orkney, and we're looking forward to keeping in touch and hoping there's an opportunity for you to come back next year.
That's a lovely, lovely place.
Oh, you'll be most welcome.
Okay, thank you.
Thanks to Ford, and thanks also to our technical team behind the scenes. Thanks to everyone at home for joining us. We've just 4 events left on this closing day of this year's festival. At 3:30, we're going to hear about whales in the changing Arctic. At 5 o'clock, we'll see a new film about the shaping of Orkney's coasts, with scenes among and above cliffs and shores of the West Mainland. At 7:30, we'll find out more about how whiskey gets its flavour. And then we round off the festival with the closing ceilidh at 8:30, to which everyone, everywhere is invited. Until then, we'll say goodbye for now.